Hey everybody, welcome back to the second half of today's lecture. If you recall, we left off with heterozygous and homozygous, dominant, recessive, and looking at alleles. And now we're going to look at some of Mendel's laws that he put together or thought up from his exquisitely done pea plant experiments. So let's look at this. The first one is Mendel's law of segregation. And so the law of segregation states that the two alleles for each trait separate during meiosis. And so during fertilization, the two alleles for whatever trait you're looking at reunite. Okay, and so heterozygote organisms are called hybrids. Okay, and so a dihybrid cross is a cross of two heterozygous organisms. That would be an example of how to use the term hybrid and heterozygous in a sentence here. That's relevant because you might have some questions. So segregation, they're separated during gamete formation, and then during fertilization, they are reunited. And that's the law of segregation. Now, the second law that Mendel came up with is the law of independent assortment. Independent. It should say assortment. I feel like there's a missing word here. Oh yes, independent assortment. Sorry, that should have been highlighted as well. And so this law states that alleles, as they segregate, do so independently from one another. So if I have an allele for a tall pea plant and an allele for green seeds, those two alleles will segregate independent of each other. So they won't influence each other's occurrence in the offspring. And so when you do crosses, you'll have statistical ratios that are unique for the green allele and unique for the tall pea plant. The two won't affect how frequently the other one appears. And this was thought to be true for a very, very long time. You still need to learn it and know what this law is, but as we will talk about in the next module, it's not 100% true anymore. All right, so a monohybrid cross, okay, is where you are doing a cross with one hybrid and one pure breeding organism. Okay, so you have a monohybrid, that's one hybrid, that's a heterozygote, and it's being crossed with a pure organism, okay, dominant or recessive. And whether it's being crossed with a pure breed dominant or a pure breed recessive, it'll give you quite a different result. So the number of squares in a Punnett square has to do with the number of alleles in the G1 generations, all right, or coming from the G1 parents. So if we're looking at one trait, you have the law of segregation, which means that each parent can give one allele and they have two. And so in a cross, you're going to have four squares with the first parent on the top, usually the mother, if I recall correctly, and it'll have the symbol for their alleles, dominant or recessive, and there should be two of them. And along the left side of the four squares, you will have the alleles for usually the father, and it'll be dominant or recessive, whatever it is. And then for the first square, you take one allele from the father and one allele from the mother, and you put them together, and that should be the allele that 25, or the allelic combination that 25% of your offspring get. And the same goes for all four of the squares. One allele from the father, one allele from the mother, one allele from the father, one from the mother, one from the father, one from the mother, and you create what's called a Punnett square, which is a cross, which is a monohybrid cross. And monohybrid crosses are usually, are exclusively looking at one trait. So you're looking at pea color, or the smoothness of peas, or the height of pea plants, or maybe in humans, you're only looking at eye color or nose length, any kind of trait that can be discretely inherited by an allelic-like pattern of inheritance, you can use this Punnett square or this cross method to try to investigate. Now, a dihybrid cross is a lot more complicated, and it involves two different traits. So, for example, crossing a green pea smooth seed plant with a yellow pea wrinkly seed plant. Okay, so the traits are the color of the seed and the texture of the seed, and you can cross those, and the two traits will sort independently, and you can make a prediction about what the offspring's characteristics will be like using a Punnett square, just like we did with a monohybrid cross. Now, obviously, you're going to have a much wider variety of possible offspring with a dihybrid cross than you're going to have with a monohybrid cross. And so Mendel's results when he crossed his plants were not exactly the ratios that you might expect, given just the allelic frequencies. And so this indicated that there was something else going on other than dominance and recessivity and independent assortment and segregation. So, with all of that in mind, and that was an absolute metric ton, we go back to the question, what is the significance of Mendel's experiments to the study of genetics? And it's a great question, and hopefully now you have some ideas and you have some vocabulary that you can use to answer this question. But if not, don't worry, you can always ask me questions. So thank you so much for listening, and I've got some questions for you. Let's get to it.